Greetings. My name is Adam Ricci. I'm going to be talking to you today about my protein that was assigned to me, and myosin. Before I get started on that, I'd like to just kind of go over a summary of proteins in general. Proteins have many different jobs. They're very important to a cell as they carry out a lot of the jobs that a cell has to do to sustain itself. Examples of that: receptors. Proteins act as receptors. They act as enzymes. They act as motor proteins, etc. That's not an exhaustive list by any means. That's just kind of skimming the surface. Regardless of what the protein's job is, they're all structured somewhat similarly. They have similar aspects that make them a protein. One of the most important things to mention about that is just the backbone. What a protein is made of: amino acids. It's a group, a long chain, if you will, of amino acids. Different amino acids with different side groups, giving each specific amino acid different chemical properties, and thus forming different shapes. And that goes into kind of what I'd like to talk to you about on this side right here. You get different structures from those different chemical interactions. The first interaction is going to be primary. That's the backbone. I already mentioned that as amino acids. The different combinations of the 20 different amino acids that we have. The secondary is formed by hydrogen bonds, and that's going to be forming helices and beta-pleated sheets. Now, the tertiary level of structure is actually made by different interactions with the side groups, forming further loops and twists and turns and giving it a more advanced structure. The final level of structure, the quaternary level of structure, kind of ties everything together. You have different protein subunits coming together by different interactions. Could be hydrogen bonds, could be sulfide bridges, etc. And that makes one big macromolecule protein. So as I said before, I was selected to do myosin. Myosin is a very unique protein. It's very important to a cell and to an organism in general. It is a motor protein, so it's the main protein that the cell uses to transport. And also, it's the main protein that allows skeletal muscle and involuntary muscle as well, but it allows muscle to move. As I have listed here, it's common to pretty much all eukaryotic cells, found largely in muscle cells. That's actually where it was discovered from its prevalence within the muscle cell. It does exhibit quaternary level of structure, and as we know, if it exhibits quaternary level of structure, it exhibits all egress as well. You can see the shape right here. This is what they call the tail, and then you have the neck and the head. The tail is somewhat used for anchoring to an actin filament, and I need to back up before I say that too. Oftentimes, we have molecules in our cell. They don't act alone, so they act in combination with something. Well, myosin is no different than that. Myosin acts with actin. Actin is the long filament, and myosin is the moving section. So anyways, this long tail, it attaches to the actin filament, and this head, or these heads, they act as an enzyme and hydrolyze ATP, changing their conformation and allowing it to actually move and reattach to actin. As it says down there, it requires ATP to work and actin filaments. There's been studies done where they remove the actin filament, and certain types of myosin doesn't even work as well or at all. So as we already talked about, it acts in muscle, but it also acts in cell transport. So if you have macromolecules or molecules that need to come in and be delivered to a certain organelle within the cell, myosin is most likely going to be the protein to do the job. As that says, it's involved in cell division. So very, very important functions for myosin. Being that it is so important and prevalent in the cell and in muscle cells, you have a greater opportunity for something to go wrong, basically, and that's where we get our diseases from, such as cardiomyopathy. You have different types of muscular disease, and it can be traced back to problems within the specific myosin units. Just as a conclusion, again, proteins are all organized similarly, regardless of their function. And I wrote here that myosin is the unifying protein. Not the unifying protein, but it's a unifying protein. And why I say that is it can be used to transport other proteins. So that was just kind of a unique thing that I thought about. Anyhow, that is my presentation. Thank you for watching. Have a good day.